Hello everyone, I'm Junha Shin, a scientist in UW Medicine. I'm going to present about a new algorithm I'm developing named Tree-Structured Matrix Factorization, or TMF in short, which is for integration and analysis of heterogeneously related datasets. The single-cell sequencing technologies are revolutionizing biological and biomedical research by providing high-throughput molecular phenotyping of individual cells at different omics levels, including transcriptome and epigenome. A major challenge here is to systematically integrate the complex dataset which are heterogeneously related, such as from a time course or hierarchically related contexts like multi-species dataset. Several methods have been developed for the integrative analysis of multiple samples like listed in the table on the right side. However, most methods project samples into a shared lower dimensional space, which might not be optimal for handling complex composition of samples. So here we developed a novel multitask matrix factorization method named TMF, which performs sample-specific matrix factorization while incorporating the relatedness of the samples. The TMF is based on NMF, or non-negative matrix factorization. NMF is a popular data analysis technique, especially suitable for sparse high-dimensional non-negative dataset. In NMF, our objective minimizes the difference between the original matrix X and its approximation from taking the product of two lower dimensional non-negative matrices U and V. U and V have a much lower dimension than the original matrix X and encode the major patterns of it, so that can be also used for clustering. Each of the factor columns of U matrix capture the latent transcription signature, which are related to the cell type specificities. So therefore, by examining these matrices, we can get out the cell cluster. To enable integrative analysis of the complex multi-context dataset, we developed a novel multitask matrix factorization method named TMF, which performs sample-specific dimensionality reduction by NMF while jointly incorporating the relatedness of the samples and species. TMF identifies the major patterns within each sample-specific matrix based on the relationship tree, which constrains gene-side vectors similarity between two different samples branched out from a parent node. These constraints could be optimized by alpha parameter of the objective function. So far, we compared TMF to several existing algorithms on three different datasets, which are representing different platforms, different samples in complex contexts such as cell lines and time course, and finally different species. We tested the performance in aspects of clustering validity and batch effect correction ability. For the evaluation, we computed three different indices or scores for both categories and added one more measurement of similarity to the known cell types of the PBMC dataset case, which was available. Although there is no universal method that outperforms other methods, TMF has the best overall rank. Next, we analyzed the clustering result of TMF and other algorithms from the multi-species dataset, which represent different stages of differentiating brain organoids. Since our expectation for a good integration in the multi-species analysis was to find conserved cell clusters that comprise same types of cells from multiple species, we observed the species composition of cell clusters. Because total number of cells for a species varies in the dataset, we defined the conservation score for a systematic comparison, which is basically a fold enrichment score of species-specific cells in a cluster. We calculated conservation score for each clustering solution from different algorithms, and the distribution of the scores exhibits that TMF captures the conserved cell clusters better comparing to the other algorithms. And next, we examined which type of cells are comprising the cell clusters identified from TMF. We firstly defined lists of differentially expressed genes, or DEGs, for each cell cluster by considering the significance in ratio of expressing cells, and then annotated the cell type identities by testing enrichment for the known neurodevelopmental marker genes. And interestingly, the conserved cell clusters identified from TMF are corresponding well to the major differentiating cell types of the lineage, such as neuroepithelial cell, radial glia, intermediate progenitors, and several types of the mature neurons. These cell type specificities could be reconfirmed by visualizing the cell type specific marker gene expressions. And moreover, the enrichment test of cluster-specific DEGS shows that the identified cell type-specific clusters are also having functional conservations, especially for the biological pathways related to the neural cell differentiation and mechanisms. For example, cell clusters annotated as cell types having pluri or multipotency, such as neuroepithelial cells and radial glia, are enriched for functions like cell cycle regulation and telomere maintenance, whereas more differentiated intermediate progenitors are enriched for neuron differentiation. Also, enrichment for cell type specific functions could be reconfirmed, such as oligodendrocyte and astrocyte differentiation for radioglia or excitatory synaptic function for excitatory neurons. So in conclusion, a multitask clustering algorithm TMF exhibits the ability to identify conserved cell clusters of a specific cell type in the analysis of complex multi-species scRNA-seq dataset. And we are planning to expand the analysis for other types of the complex datasets such as multi-omics dataset. Thanks for listening.